Welcome to our presentation on the regeneration of traumatic and non-traumatic injury in brittle stars. The nature of our experiment is to determine the elements of regeneration in brittle stars, which are an organism that specializes in regenerative capabilities. Using brittle stars as a model, we used a sampling system involving microscopy and the use of sterilized toothpicks, which we use to swab the sites of amputation and examine cellular processes under the microscope and track the regeneration process or the lack of regeneration process over the course of 2 weeks. Our care procedure included a daily chemical test of nitrates, nitrites, ammonia, water hardness, and pH. We conducted a partial water change every 3 to 4 weeks, where we remove almost half the water from the tank and pour new salt water back to replenish the water and remove contaminants.
In this picture, you can see the habitat that we set up for the brittle stars.
Our experimental procedure had 2 different procedures involved in it. The traumatic injury procedure was for a brittle star that received a traumatic injury, which was provided using a increasing amount of tension at a certain length across the arm. In this instance, we used approximately 2 inches from the tip of the arm towards the base, and by applying greater amounts of tension, we were able to separate the arm, creating a traumatic injury, which then we swabbed with a sterilized toothpick and plated. We took multiple samples across multiple days, and some samples we used the swab and used deionized water and centrifuged the sample for a more consistent homologous cellular sample, and we were able to see different cellular communities under the microscope. And in the non-traumatic procedure, the only difference was the use of a scalpel to make a clean cut approximately 2 inches into the arm of the brittle star, which provided a more subtle damage to the brittle star, which induced non-traumatic injury.
The safety protocol included we wore gloves to prevent contamination of any organisms or contaminating the tank water, and any biological material was disposed. And there were no dangerous biological agents in the brittle stars themselves. So,
and test kit chemicals were handled according to their instructions. Here you can see the test kits being used to test the water hardness, pH, etc.
We observed that the non-traumatic samples were more see-through and had more cell communities, while the traumatic samples had less cells and were more opaque. The brittle star inflicted with non-traumatic injury showed signs of regeneration, but there wasn't enough time to collect data concerning the actual growth of the arm. And the time period was short, but over the course of a week, the brittle star inflicted with traumatic injury had little to no progress in regrowth based on the observations and measurements of the length of the arm.
Here you can see a community of cells collected from a sample with a non-traumatic injury. These cells are plentiful, and you can see they are coming off the sides. Our results were limited by the amount of data collected. We only had 2 weeks. Months of data would have been more optimal, but tissue samples were collected thus far, and they described some element that points to or inhibits the regeneration process, and that takeaway is the nature of the injury threatens the primary functions of the brittle star, which would mean that there's some trade-off here where the life of the brittle star is put in first priority and the regeneration process is put on hold by some other element where it would normally regenerate where it was a non-traumatic injury. Jack, could you describe the methods we used?
Methods outside of microscopy include lysing, and microscopy is limited to surface level cellular analysis, while lysing opens cellular elements, which, when compared to elements in a non-traumatic versus traumatic injury, can reveal further information regarding what kind of factors decide what happens during the regeneration process. While our research was inconclusive given the 2-week time frame, there were observable trends that pointed towards some sort of element that in the traumatic injuries that caused the process of regeneration and regrowth to be inhibited, and the nature of the factor that inhibits their ability to regrow and manufacture new cells to produce a blastema is yet to be identified. However, through further data collection and new methods for cellular analysis, there is potential to isolate the factors responsible for regenerative inhibiting in the face of traumatic injury.
And following through with the lysing methods might produce more cumulative data that can point to the specific method or specific element that's responsible for the inhibition of regeneration in the brittle stars. And there are additional unforeseen factors in our experiment, such as the chemical composition of the water, which was maintained throughout the chemical tests that we did, and concentrated bacteria that broke down the unwanted byproducts and nitrates of the tank's natural ecosystem. However, the sea life existing in the tank consisted of small crabs as well to provide a more natural habitat, but the unforeseen factor was the death of one of the crabs, which created an excess of ammonia as it decomposed, potentially detrimenting the health of the tank for a few days, which might have caused inconsistencies in our data. And one more thing, the brittle stars were ordered from the Carolina Biological Supply website, and thus were outsourced from Florida, providing varied species. However, we used the two related species to maintain consistency in our experiment. Thank you for listening to our presentation. All right.